Okay, I'm glad to be here again. It's a lot of pressure now because this last talk before the evaluation. So if I really screw up, this will color the whole impression, and then I get a lot of bad feedback from the course instructors. So I'll do my best. I hope you found the course informative and useful. It's an intense course. It was also the first time in this format, so in some way you were guinea pigs, but I heard some feedback, and I think it went, my impression anyway, that it went quite well, but we'll see from the evaluations. So I would like to make some comments about the reproducibility of research results, specifically in the field of proteomics. We now discussed a whole week a technique which certainly has been designed initially to be a technique which has a high degree of reproducibility. And this was actually the initial intent. And so rather than finding as many proteins as possible, was to find a way to do measurements as reproducibly as possible. And I would now like to give a little bit of background, why this is important in experimental biology, and then where we are, and then with this and show some applications. So this is an outline. We need to be able to generate reproducible research results in general, and certainly in the field of proteomics. And I think we need to do this for political reasons and scientific reasons. I'll expand on these points. This is just an overview. Then I would like to address 3 questions with some data. And the question is, if 2 or more people have the same proteomic data, can they generate the same results? The second question would be, if 2 or more people have the same sample, can they generate the same proteomic results? And the third, if 2 or more people do the same experiment in a cell line, do they get the same result? So these are very, of course, at some level trivial questions, but they're actually very profound and actually complicated. And I think my take-home message would be that we have made, through the techniques you learned in the field of proteomics, substantial advances towards reproducibility in terms of data generation, data analysis, and the research results coming out from proteomic data sets. But it does not mean that that the third question can be necessarily answered with yes, because there's many other reasons why an experiment that someone is doing, or if 2 people are doing ostensibly the same experiment, end up with different results. Okay, so why is it important to have reproducible research results? Of course, this is the bedrock of experimental science. We believe that if some person or one group is doing an experiment and another group is doing an experiment, the same experiment, as well controlled as possible, that they should come up with the same result. If that is not the case, then either someone screwed up or there is something happening that we don't understand. Many clinical or other biomarker studies depend on the comparisons of many samples. You cannot find a biomarker from 1 sample or from 5 samples. Usually you need dozens to hundreds of samples. And to do a meaningful comparisons within a cohort, of course, you need some form, preferably a very high degree of reproducibility, both on the level of what you identify in terms of which molecules, but also how you quantify. And then we alluded to that a little bit on Monday, that systems biology studies relate a lot on machine learning, correlative statistics, and so on. Basically, we work with large data sets where many samples of a cohort are measured, and we try to extract some information, how the system behaves under certain conditions. And of course, here the data would also need to be reproducible. So, and I think this is now an important political issue. Perceived real or poor reproducibility is usually construed with poor competency of the researcher, the scientists, or worse, as someone basically who cheats. And so this is an important political issue. And in fact, we as scientists should worry about that greatly, because there is a whole literature now being published. It's percolating through scientific journals, but also the popular press, newspapers, that inherently when people try to reproduce something that has been published, it is hard to do. So these are some studies that show that a fraction, small fraction, surprisingly small fraction, of published science articles are reproducible. And the rest, of course, are not. And so this is usually then interpreted that either we don't know what we're doing as scientists, or we're cheating. And I think neither, I mean, well, that's one of the themes that I'm trying to come up with or follow through here. Neither of this conclusion is necessarily correct. I'm not saying that everyone is highly competent. I'm also not saying that no one is cheating. This unfortunately happens. But for the most part, I think that what happens when something is not reproducible, that there's underlying variables which we simply do not understand and cannot control. And then the results will be different due to the fact that some factor, confounding factor, is applied to in someone's research laboratory and not in the other, and then it leads to different results. So finding these factors and eliminating them would be an important issue in experimental science. And this is not just nice for someone writes the thesis, that they know these results are reproducible, but it has a lot to do with science funding. In many countries, fortunately not so much in Switzerland, but for instance in the US and in other countries, science funding is under big pressure because a part of the society is inherently science opposed, and they only look for reasons to basically eliminate or reduce science funding. And there's nothing easier for someone to say, we want to reduce science funding, than saying, well, look, what they're doing is basically irreproducible, it's crap, it doesn't work, they don't know what they're doing, so it's a waste of money. And I think it is important for us as scientists to say it is difficult, but we're not cheating, we're not incompetent, but we need to learn basically why things are not working or why things are working. And we also think we need to make an effort to make things more robust. And I think this is one of the attempts with this course, is exactly to do that, to try to convey information to a group of motivated students and scientists, that they would acquire skills which we believe will help them to do certain types of experiments more reproducibly than maybe in the past. Okay, so now this is being discussed a lot in the journals, specifically Nature Publishing Group has a whole string of discussions and articles about this reproducibility issue, and they did actually a very good job in saying this is not a single item issue. They break it down into various steps. So they say, for instance, experimental design, reagents and drugs, data acquisition, data statistics, validation, and so on. And so this is very good, because this would allow us to try to see where in the process from the experimental design to the published research result, where are there problems? And if there are problems, how can they be alleviated? So I will come back to some of these points throughout, because I think now in proteomics we have made some progress, I think fairly significant progress in some of these areas, the data acquisition, statistics, and validation of data sets. We can confidently say, if someone is doing an experiment competently, that the results are quite reproducible. And we'll show some results of that. And then there's a number of journal articles who address this issue. So now I would like to make a general comment about the issue of reproducibility and variability. So we try to do measurements, and we try to make measurements that are basically the same when we do it again. So if two people do an experiment, let's say on a cell line or a mouse or a yeast strain, and we do two groups do the same measurements, we think there should be no variability. Of course, this is not possible, because there's always some technical variability, but we would like to say if two people do the same experiment, if the technical variability is under control, they should come up with the same results. And in biology, this is actually not necessarily so, because there's an underlying enormous biological variability which is clearly real, which we should probably try to exploit, but which usually is not considered. So I'd like to make a difference here, distinguish engineered systems and biological systems. So if you're an engineer, let's say if you work at Toyota or Volkswagen or so, you will work towards engineering a car, and this car will be optimized. It will be tested. If it has flaws, these flaws will be eliminated. Maybe it makes a noise. This noise will be eliminated until somehow a design has been reached which is considered satisfactory or optimal. And then millions of these identical specimens are being produced. They should be all identical. They should use the same amount of gas. They should have the same gas mileage. They should make the same noises. They should be identical. So this is not so, distinctively not so in biology. So there is no universally optimal solution. Biology is not driven by coming up with an optimal design or even an ideal design. And then there's many copies of that made. Biology lives from the specimens that they're different. And in fact, variability is essential for a survival of a species. We believe that. We know that also from experiments that are being done. For instance, in agriculture, when uniformly engineered plants, for instance, are seeded out in big areas, they're prone to being affected by a disease, for instance. And then if they're all identical, this disease will affect everyone in a single way and essentially wipe them out. And the reason that biological species survive is that there is a great biological variability, and we can look at each one in here. Everyone is different, and this has some disadvantages, and it has some advantages for the species. It has advantages. For the individual, it may be not optimal. Someone may be not able to run up a stair like as fast as the other person. So in terms of athletics, one may have advantages, but this athletically very astute person may have disadvantages in some other field. So I think this is an important difference between engineered systems, biological systems, which is oftentimes not considered. So in biological experimentation, we should be able to distinguish between poor reproducibility, basically noisy data, and essential biological variability. And oftentimes, this is not actually done. So what we learn as students if we get into experimental biology, we all learn to do a controlled experiment. And what a controlled experiment basically entails, it has a certain recipe. We take, for instance, a mouse or a yeast strain or a cell line, and we control as many variables as we can. So, for instance, we would keep the strain constant. We would keep the age of the mouse constant throughout an experimental design. We would even go and look exactly, take the mouse at the same time of day to do some measurement on it, because we know it has a 24-hour cycle. We would control the food state, the temperature, maybe even the microbes in the cage. Maybe we would eliminate all the microbes in the cage to eliminate one confounding factors. So we essentially establish a highly controlled environment, and then we do one variable. For instance, we would take this mouse with all these things controlled, and we would change one variable. For instance, we would add a drug, or we would delete the gene, or we would mutate the gene, whatever the case, and then we do measurements. And then from these measurements, this may be proteomic, maybe genomic, maybe something else, we would make a finding. We would, for instance, say, if you add this drug, this particular pathway is inhibited. And then we would write a paper and generalize this finding and say, in the species mouse, if we add this drug, then this pathway is inhibited. So up here, everything is fine. This is certainly a good idea to keep things under control and have low number of variables. It is good to do a targeted experiment in terms of inducing a change. But here, the big flaw of this is that we then do a generalization from this one mice under this condition at this time of day to make a general statement. For instance, saying this gene is an oncogene, and it may be an oncogene in some mouse strain, but not in another. So to illustrate this, we realize that mice, exactly as humans, are different individuals. They have different genomes, and these genomes will lead to some different traits, different phenotypes. So many of these strains have been mapped out genomically. In many mice, there have been experiments being done, and then there is clearly the observation that these mice do not necessarily behave the same way under any condition, under any treatment. So there is no reference mouse, there's no ideal mouse, there's no ideal person, there's no reference person, there is no reference state. So all results that we measure are conditional. And mice are different and likely act differently if you were to do experiments. So this conditionality is usually not reported in journals, and because it is important, as I'll just show, it means that what is presented as a finding with no conditional information attached will be true for some conditions, but not for others. Okay, now we know, of course, that there is a relationship between our genes and between some of the appearances. So this is actually a picture from Hiro that he produced for a talk and a study he generated. And I just use this here to say we can measure now very effectively single nucleotide polymorphisms, basically the genomic variability in a human population or a mouse population. And we can relate this SNPs to phenotypes. So we might, for instance, ask, how does a specific genetic locus, if mutated, affect the size of these individuals? They obviously have vastly different size. We would assume that this has to do with genetics, not only maybe, but that there's a link between here, genomics, and the phenotype. And this is referred to as quantitative trait locus mapping, where we try to find a linkage, as an association, a statistical linkage, between something that's observed phenotypically and some genetic variant. How mechanistically this works, how a particular SNP, if we find such a SNP that affects the phenotype, how mechanistically this works is a different question. We simply establish an association. I will then show an example where we can use data acquired in a suitable cohort to make a link between association, so basically saying there is a link here, and some mechanistic understanding. But now, if we go into a population of mice or a population of humans, there is enormous variability. And it is very difficult to make sense out of this genomic variability and to relate it in an outbred population to phenotypes. You need enormous numbers to do that. And so some of the genome-wide association studies, which try to establish these linkages here, SNPs being millions of SNPs, phenotype being a complex disease like diabetes or so, many of these studies now involve hundreds of thousands of genomes. So this is not really tractable for an individual laboratory. But it is tractable to some extent if one would generate strains or basically engineer populations of controlled variability. So this was done, and this is now slides from Evan Williams, whose work in our group is a postdoc who works with this cohort. And the intent here was to generate mice, strains which are inherently homogeneous. So you have multiple copies of each mouse, but each mouse is different from the other, so that mimics kind of the population. But they're not as different as from each other that outbred individuals. And so these mice were generated so that they have a number of sequence variants, and hundreds of phenotypes were measured. So these mice were generated by crossing two parents, a mother and a father, from two laboratory strains, And then through genetic crossing, which I don't want to discuss here, strains are generated, more than 100 strains, which all have in common that they are inherently within one strain identical, so we like identical twins, and that between strains they are different, but the genome from each one of these strains is composed of various proportions of the one parent or the other. So the variability is constrained, not as wide as a general outbred population. So now why am I talking about this? Because we now have a resource of basically like a gigantic family, with each family member having multiple copies that can be used for experimentation, and the variability is somewhat understood and of course can be measured. So we have loci, of course, on each locus. There's either the allele from this one or from this one, but not a different allele. And these mice have been through a consortium project, been phenotyped with large numbers of phenotypes. And now this allows us to ask, how does the measurable phenotypes that can be measured on these mice, how does this relate to the genomic alleles in specific loci? So this shows now some examples that Evan pulled out. So this would be a particular phenotype which is interesting for athletes. This is the ability, the maximal ability a person, in this case a mouse, can process oxygen. So Tour de France is starting tomorrow, I think, or maybe tomorrow. And you can be sure that if you were to measure each one of those riders, they would be very high, have very high VO max. So they have the ability to burn a lot of oxygen for a long time, and that gives them the enormous athletic performance. Other people do not have that, so they can train as much as they want. They will never make it to the Tour de France, and certainly not, they will certainly not win it. So here in the mice, it's the same. So here we have in the mice, the two parental strains were grown with two food conditions, normal food and high-fat food. And this is the one parent and it's the other, and they have roughly the same VO2 max if they are on a normal diet. If they have high-fat diet, are getting fat, they have lower VO2 max, which is also understandable, but they go down to a different degree. So there's a genetic component that says they have roughly the same VO2 max, but they react to high-fat diet in a different way. There's another phenotype, which you don't need to really belabor what it is. It's a medically used phenotype. And we see here that the two parents have a different level of this particular enzyme in the blood. So clearly there's a genetic determinant which says this is low, this is high, but when we put them into high-fat diet, they all go down to roughly where this one is in level. So the level of this one here, parent, is affected. The level of this one is not affected by high-fat diet. So now you might say, okay, this is maybe somewhat interesting, but why am I talking about that? It comes back to this variability, biological variability in a population. Now we have here the two parents, but now let's see what happens in the strains. And the fascinating thing is that when we now see, these are the VO2 max measured from these strains, the segregants from this initial cross, we see that they segregate in a vast range of VO2 max values, numerical phenotypes, and that the two parents here are at the very end. So you would normally expect, if this was a Mendelian trait, you would expect that the children would be either like the one parent or the other parent or somewhere in between, but not outside the value of the two parents. And we see here most of the segregant strains have a value that's far outside the value specified by the two parents. And this is the same here. This is serum alkaline phosphatase. This is the one parent. This is the other parent. And some of the descendants are within the range specified by the parents. and a lot of the children, the segregants, are outside. So now this immediately points to a very complex relationship between the locus, the allele at the specific locus, and this phenotype. And now if you want to investigate this, coming back to this topic of reproducible research results, we need to be able to distinguish between biological variability and between technical variability. So these are not necessarily very big differences here between the strains. And if you would like to do something like quantitative trait locus mapping, we need to confidently say that this mouse here has a value that is different from this mouse here. Otherwise, if they are within the experimental noise, we of course cannot see any signal, and we basically correlate noise with noise. So this is the technique that you learned here, DIA, and also selected reaction monitoring, the kinds of precursor targeting technique. They have been clearly shown that if they're applied properly, they can be so precise across a cohort that we can distinguish the technical noise from biological noise or biological effect, even if this effect is not very large, like we would expect here. So how do we know that? We know that if we do repeat analysis, and this is something which should flow into the experimental design of any such study, that we will need to do repeat analysis at the technical level to see what is the variability that is achieved technically, and then if we do a biological experiment, let's say a perturbation experiment, we would be able to say the variability achieved in the biological comparisons is larger than the technical noise, and therefore we see a biological relevant signal. So this is such a graph. This would be coefficient of variation. This would be technical. This would be replicates here, the blue, and they basically have a certain. Each dot is a protein that would be measured, let's say, by DIA, and then so this is the variability. We would read that and say this protein here has a technical variability measured by repeat technical analysis that is very low, and then some proteins have a higher technical variability. But the same proteins will have, in the biological samples, a larger variability, and we conclude from that is that the difference, that because these curves diverge, that we are able to see biological signal, distinguish this from technical variability, and that we can see if something biological is going on here, we can actually detect this. So this is coming back to a point I made on Monday with the simulations that Ting, the student from Olga Witek's laboratory, generated. I don't know whether you may remember this, where I said that it is more important to have protein measurements on fewer proteins, but that are precisely measured, than a very large number of proteins that are noisy, if we want to make such associations. And this is exactly the same issue again. If we had this blue curve out here, and basically overlapping with the red curve would mean we have so much technical variation because the molecular measurements are so noisy that we would never see a biological effect. And by applying these targeting techniques, we can confidently say, by this analysis of repeats versus biological samples, that we can actually see biological signal. So now I would like to show what we can learn from that. And this is, again, work from Evan Williams, who exploited these BXD mice strains, and he analyzed liver samples from these mice all across different strains and under different conditions of food intake. And what we like to do, we have the genomic sequence of these mice, and we know the genomic variability, and we would like to correlate genomic variability with observed phenotypes, such as body weight, metabolites, and so on. So we can basically go in an environmental perturbation axis, a genotype axis, and the goal is to find out how is a particular measurable phenotype generated and how is it affected, to what extent is it inheritable, can we reduce it to specific genes or not. So I don't want to show a lot of data, because I want to get back to the proteomic landscape. This is actually a proteomic experiment, and here he observed by doing actually limited measurements. He has now repeated this with SWATH MS, but these are data from an earlier study with SRM, but the picture is the same, it's just lower numbers. He observed total 44 cases where the abundance of a protein could be correlated with the presence of an allele. So if you have, let's say, one parental allele, this allele would lead to a high level of the protein, and if the other parental allele is present, it would lead to a low level of expression of the protein. If you have a sufficient amount of measurements over sufficient mice and conditions, you can assign a statistical value that indicates the likelihood that this association is secured. So you can say this particular allele at this locus affects the abundance level of a protein. So this is a number of these were found. With SWATH data, much larger number was found. And then I don't want to comment about these others. And then the same was done also with the transcripts. So we always confronted in proteins with statements like, is the protein just retracting what the transcript is doing? And these data clearly show it's not. So we find actually with a similar number of QTLs, so links between the transcript and the phenotype, like the link between the protein abundance and the phenotype, but they're differently distributed. So the protein has a much higher number. The transcript has a much higher number of cis QTLs. That means the locus, the allele itself, affects the abundance of itself, as opposed to trans, where the allele affects the abundance of some other transcript. And so this is different from proteins. So in proteins, it seems much more divergent, more indirect regulation, because only a third, roughly, of the QTLs was cis, and about 2/3 was trans. So with this points to a different level of regulation, that the relationship between the abundance of a transcript and the genomic locus seems to be closer connected to itself, and the further you go out in terms of protein expression and expression of expressed information at the protein level, then it seems to become more complicated, more indirect. Okay, now this is simply associations, and so Evan was able to find here a number of loci which affect the abundance of a particular protein. It's simply a statistical association. If you do enough of these measurements, you can say you have a population of individuals. They're genetically different. Here now in these mice, the genetic variability is constrained, but we can associate a particular allele with an abundance of a protein. So this can be done, and of course it's interesting because it establishes a link. But now what about mechanics? We are interested as biologists usually to reach some form of mechanistic understanding. And this is a very nice example from integrating data at various measurement levels, specifically proteins and transcripts, into a mechanistic model of a process. So this requires, again, that the measurements are done at each level, here metabolites and proteins, at sufficient precision and accuracy that we can see biological signals in the technical noise. So now what came out? One of these QTLs, so this link between a genetic locus and the protein abundance, linked to a protein which is the last enzyme here in a pathway that degrades branched chain amino acids like lysine and basically degrades them. As typically is the case with metabolic pathways, this degradation doesn't happen automatically or in one shot. It happens by a sequence of enzymatic steps where the amino acid is reduced and processed from one step to the next, always with an enzymatic reaction. You probably everyone learned that in basic biochemistry, metabolic processes, and they all work like that. So this is one of them. So now what Evan was able to measure is intermediate metabolites of this enzymatic cascade, and one is called amino adipate, and this is another one which was measured. So now we can basically measure quantify, we have a link between the genomic locus, which codes for the enzyme down here, that carries out the step. We have a link between the enzyme and its biochemical products. Now we can measure these two things and correlate them to each other. So what we see here is whenever this is the liver enzyme level, and this is the metabolite here, we see that they're inversely correlated. So if there is a large amount of the enzyme present, then there's little of the metabolite here, and the reason, of course, because it is processed. If you have a small amount of the enzyme present, then there is a large amount of this metabolite because it is being generated upstream, but it is not sufficiently fast processed. So you can think of this like a water hose, where you have a constrictor at the bottom, and when you constrict the flow by having less of the enzyme down here, then upstream it piles up, and if you have released the constriction, you have more of the enzyme, more flux happens. So this is clearly coming out of the data and basically explains that a genomic variant leads to the control of the abundance of an enzyme, and this is then measurable or translates in the level of this particular metabolite in the cell. So it's a genetic, mechanistically explained control of a metabolite in the cell led back mechanistically to a specific genetic locus. So you might say, well, this is interesting, but it turns out that this amino adipic acid, this metabolite over there, is being identified in biomarker studies, in epidemiological studies, as a biomarker for diabetes risk. So basically, what now this study makes is a link between a genetic variant and a particular locus, mechanistically linking this to an enzyme abundance perturbation, to a metabolite abundance perturbation, and which constitutes or is associated with the risk of a disease. And more work that was carried out, which I don't want to explain here for reasons of time, showed that while this was done in mouse, these QTL analyses were done in mouse, that this has also bearing for human biology. So this is a nice example, I think, that shows that only if we can do reproducible measurements of proteins, in this case, and also metabolites, over a very large cohort, and we can do these measurements fairly precisely and with low technical variability, can we start to undertake association studies. And then using the backdrop of biological pathway knowledge, we can at times, this is of course a special case, it will not work every time, we can use this information to learn something biochemically, even all the way to explaining mechanistically a disease biomarker. This is only possible because we use accurate measurements, reproducible measurements, and also because we take actually advantage of the biological variability that's inherent in these mouse cohorts. If every mouse was exactly the same, we would of course never find that result. So in addition to these issues of reproducibility which I mentioned before, I would now add that understanding for biological reasons, we can understand biological reasons for variability, which uncovers interesting new biology, and highly precisely measurement in a suitably constructed cohort distinguishes between poor reproducibility and biological variability, leading to new biological insights. So this is scientific justification for establishing techniques where you can measure the same thing over many conditions with high precision. So now with this I wanted to illustrate why we need, not just for political reasons, but for very good biological reasons, establish methods which are very highly reproducible and can measure lots of stuff so we can do association studies. Now I'd like to ask the question, coming back to proteomics, the technique we learned here, I'd like to ask if two or more people have the same results, proteomic data, and these are SWATH data that were generated, can they generate the same results? This is addressing the question of data analysis. It oftentimes happens that people do have samples, they do the same measurements, or that the same measurements are carried out in different laboratories, and then the results are compared and it doesn't look very good. So it was initially a very important step in this development of this DIA technique to ask if we have a data set which has a ground truth in its composition, but also in its quantitative relationships of proteins, can the software tools that we have available find the same results from a specific data set? So this is a study that was published last year, was headed by Pedro Navarro, who used to be a postdoc here, and by Stefan Tenzer and Ludovic Gier, who was, I think, also helping, contributing to the course. He generated the samples and actually had a lot of input in the design. You already know this data set because we worked with it basically all week, so I'm not going to say a whole lot about it, because it was already discussed. I just want to make a few comments. So just to remind everyone, these were samples which were composed of 3 different proteins from 3 different species, and these 2 samples were mixed where the proportions of the 2 species were different, so that we were comparing A to B. Then if you looked for coli proteins, they should be in a ratio of 1 to 4, and so on. So you all know that. I don't need to belabor that. These samples were then acquired in various instruments by DIA and in various acquisition modes. And then this data, the original results generated, were analyzed by these 5 software tools, which have been independently generated by different groups. So you know all these software tools probably better than I do by now. And then the results were compared. So this is simply the experimental design. These were analyzed, these samples, and then multiple runs were done, and then the data sets were distributed. So I don't want to spend a lot of time because you discussed this data during the week, but I just want to make a few pertinent points. The study had 2 iterations. One iteration was that the data were analyzed by the authors of these respective software tools, basically with the tools as they were at the beginning of the study. And then these authors of these software tools got together, they discussed the performance, and they discussed the comparisons. And I don't remember exactly the details, but I think most of the software tools, if not all, found that by doing this comparison, this benchmarking, and if they got together and discussed it, could actually improve the performance of their own tool, because they found maybe a bug, or they found some other way that they could optimize the output. So from a sociological point of view, this is an interesting exercise, which I think should be repeated more often. That is not the question, is not which is the best tool. The question is that we wanted to address in this study. Can the tools that can be applied freely in the community achieve results which are actually accurate and reproducible? And when we actually went through the review, this came up repeatedly. Reviewers said, well, this is not an interesting study because we don't say which tool is the best. And we had endless arguments to say, look, this is not the question, which is the best. The question is, are these tools reproducible and accurate? And do they give the same results? And because from a community point of view, from the point of view of reproducible research results, it is not important who is the best. It's the question that if two people analyze the same data with different tools, do they come up with the same results? And so this was a lot of discussion with reviewers and editors, and eventually this was accepted, but it was a very difficult process. So the first point I would like to make is that each one of these tools, through the iteration, discussion, and the benchmarking, actually improved in its performance. And the second point I'd like to make is eventually, at the end, they performed all very well, and equally, or not equally, but similarly well. And we all see for each one of these tools, we see that when the signals get low, here, then we see this trumpet plot I talked about on Monday, So then the technical variability simply becomes bigger. It's nothing to do with the tool. This simply has to do with the data acquisition. If you measure low signals in a noisy background, the signal to noise becomes more variable. Then, of course, the question was also not just how are they quantitatively accurate and reproducible and show the same results, but do they also basically show similar parts or identical parts of the proteome? And this is a comparison. I don't know whether this was already discussed. I don't really want to belabor that, but the fact is that irrespective of the tool that is being used, they see the vast majority of peptides they would identify identically. The biggest outlier in that regard is DIA umpire, simply because it does not use the same a priori library-based searching. It generates the libraries it uses to search the data by itself. And so there is, of course, if the library is different to start with, then the results are also somewhat different. But even, so for instance, DIA umpire might find peptides that are actually in the sample, but not in the library, because then the other tools, which are library-based, would not find this peptide, but umpire would find it because it adds it to the library, and this was actually investigated in the revision. And when then peptides that umpire found were added to the library, then the other library-based tools also tended to find them. So we don't need to worry really about the numbers here so much. The main point I would like to make is that now we are in a domain that if two or more people have the same proteomic data acquired by this DIA technique, and I should stress that this is not true for other acquisition techniques, with exception probably of SRM, and if they use any one of these tools that were participating in this study, they can be reasonably sure that they will generate data that are very similar and therefore reproducible to some other laboratory which would analyze the same data with another tool, or even if they analyze it with a different tool. So this is at the level of data processing and analysis. This DIA technique, thanks to this community effort, which is actually admirable that everyone got together and was willing to admit that maybe their tool needs improvement here and there. And it's not the question who is the best, but do they achieve something which is helping the community to generate reproducible research results, that this goal has been substantially advanced. So now I would like to ask the next question. If two or more people have the same sample, can they generate the same proteomic results? So again, this is on Nature's list here, data analysis, acquisition, basically data acquisition. So again, we did a community effort, and this was led by Ben Collins, who was also doing a lot of work in this course, and by a scientist from ABSciEX, Christy Hunter, and also with a lot of help from Yansheng Lu, who is a postdoc in our group. The experimental design was that we took a lysate of a human cell line, digested it and made lots of it, and into this cell line we titrated a number of 30 isotope-labeled peptides at various abundances. So there were a number of sample set was created, where these synthetic peptides were basically going through dilution series, and then these samples were sent out to various laboratories, in fact 11 laboratories who agreed to participate. They used the same machine, acquisition machine, a triple TOF mass spectrometer, and they used everything else, and they agreed on acquisition windows, but everything else was basically left to them, like exactly what column they used, what pump they used, and so on. So we did not try to specify too much, but to basically say, are we able, in a realistic experimental situation, where different laboratories acquire data from a sample, can we acquire results that are comparable? So everyone agreed to run these samples in a certain regimen over a week. So on day 1, let's say Monday, Wednesday, and Friday, they would commit to run 7 samples in a certain order which was specified. And this order was designed to allow, and then in the data analysis, to ask how reproducible is the acquisition within 1 day in a laboratory, across a week in a laboratory, and across laboratories. And we wanted to ask 2 questions. Do we detect a consistent set of proteins across laboratories? This would be kind of the big data question. If people analyze the same results, would they come up with roughly the same proteins identified? And the other was a more analytical chemistry component of the study. Do they show similar dynamic range, limit of detection, and limit of quantification, and so on? And this is, of course, based on the isotope labeled peptides, which are titrated into the sample. Okay, so these are some results that came out. These numbers here refer to the sites. Each site had 3 times 7, 21 analyses, and then the data were sent over here, and they were analyzed to the pipeline that you basically learned this week. So the variable in this whole study is not the sample, it's not the software, it's the acquisition. So what we see here is that internally the numbers are actually remarkable. So there's about 5,000 proteins were identified per site. Some had very excellent performance, very stable, very high. Some had some more variability. In some cases, the sensitivity of the instrument dropped down a bit, and this is, of course, real-life experimentation in laboratory. You cannot control, with every injection into the instrument, the performance goes down slightly. But overall, they performed actually quite remarkably similarly, and this simply says that they identified a similar number of proteins by their acquisition. This does not say anything about reproducibility. But this blue curve says something about reproducibility because it is an accumulation curve and basically says that up to here that many proteins are identified. And then when you go with all the runs up to here, then that number of proteins would be identified. So the curve you appreciate is that this curve is essentially flat. And so that means that if you add the data from this site here or from this site here, they do not discover any new proteins or very few new proteins. So basically what they discover is what the others also discover, and that means that the data are highly reproducible. So this would indicate that over these more than 200 runs, the proteins identified in the first run are to large extent, of course not completely, but to a large extent also found by other sites, and this is a fairly indication of a fairly reproducible acquisition method. We actually learned in this context, which delayed this publication for almost a year. We learned that when initially from the analysis was compiled, through these 200 and plus runs, that this curve started going up. And so we then investigated this, and I think this is a great accolades should go to the team that investigated this, was willing to hold this paper back till this was resolved. And we concluded eventually that the slip up here, the sliding that we initially observed of increasing numbers by more data added was an accumulation of false positives. And this was then investigated mostly by George Rosenberger, who was a student here, and he and Isabel actually developed procedures that investigated this, and now there's procedures to control the FDR on large data sets quite effectively. So this is an important step for achieving reproducibility. This shows the data completeness, so this would be about 80% of the, it basically shows how many proteins were shown reproducibly across all sites. And we see that for some proteins, of course, down here it gets also leaky, but there's actually a sizable fraction of proteins that are seen in most sites, and this should be seen in most sites because the sample is the same. So this is certainly not perfectly reproducible, and of course some of this reproducibility has to do or of these missing values have to do with sensitivity of the instrument that it can achieve, because if the instrument is not as sensitive, then the lower abundance peptides will simply be in the noise. But the ones who are, there's a remarkably large fraction of proteins which are quite reproducibly seen. So then we wanted to ask, what about the quantitative performance? It's one thing to say, well, we see roughly the same proteins. The other is, as I tried to explain in this introductory statement, is we need to have precision in measurements, and precision, of course, also means that if it's very precisely and accurately measured, we can compare results across studies. So this is basically a dilution series of these peptides, and each dilution series of concentration of peptide on the column, and then against the peak area. So this is what one normally would do. In dilution series, one adds half of the sample, and then the signal should go down by half. And so this gives a linear range of basically dilution curve. And these are dilution curves that are generated from the raw data. And these are dilution curves generated after simple mean normalization, because not every instrument yields the same signal for, let's say, 1 femtomole of peptide, because maybe the detector is slightly dirtier or older. But with a simple mean transformation, basically shifting these curves on top of each other we can do that because we have thousands of these peptides which could be used for calibration purposes. Then they go together very tightly. So these are the performance of the different sites. So we can claim that if these data are acquired with a fairly standard acquisition technique, and then if there is a simple batch effect or sensitivity correction of the instruments, each one of the sites generated for these peptides fairly nice dilution curves with similar dynamic range and similar limit of detection, although the limit of detection is not identical. You see some go further down because the instrument was more sensitive. So now also, just as a side note, we have the ability, when we generate this DAA SWATH data, to compare the detectability and quantification between measurement at the MS2 level, basically by the integration of the peak areas of transitions, as you learned this week, and by following the same peptide at the precursor level. Because every time the mass spectrometer goes through a cycle, it also, a SWATH cycle, it also acquires MS1 peptides. And of course, MS1 signals of the peptides. So we can find these peptides, and this is some examples. There's also on top is always the MS1 detection and quantification. This is the same peptide, this is the sequence, that is shown at the MS2 level. And we see clearly that when the MS1 level gets fairly noisy, and through filtering out of noise, by the fact that we look at the fragment ions and not the precursor, and therefore noise is reduced, we see here very nice transition groups for this particular peptide, whereas the precursor ion of this peptide would be already in the noise, could not be quantified. Same here, and same here. So we believe, and this was then borne out by some more broader analysis across the various sites, these are the 11 sites, that if we quantify these peptides, isotopic label peptides across their dilution series, either from exactly the same data acquired in the same instrument in the same site. If we quantify them by MS2, by the SWATH analysis, or by MS1, this is about an order of magnitude different in limit of detection. Okay. So to summarize this part, I think that right now the technique you learned this week is an intensely validated and well-supported mass spectrometric technique, and it has been benchmarked and validated in the acquisition. It has been benchmarked at the level of the software tools, and it has also been benchmarked against ground truth against manually curated data sets. And so all these steps have been published, and that's why I think it is a well-supported technique which is relatively straightforward to implement in the laboratory and has the very high potential to generate actually reproducible research results within the limits that it can do, the limits of detection, limits of quantification. But within these limits, then it is, I think, currently the most strongly benchmarked proteomic technique. So now I'd like to this running out of time, but I'd like to come to last question, which I posed at the beginning, and is if 2 or more people do the same experiment in a cell line, do they get the same results? We could exchange cell line with mice, with humans, and so on. This comes now back to the question. If you do an experiment with a particular sample, how representative is this sample for other cells, strains, or individuals? So this is work that was done by Yansheng Lu in the laboratory, and it basically addresses this area here, reagents, cells, because you could appreciate that if you take a different mouse or a different cell line and you ask how is that cell line reacting, that you might get different results because of the issue of biological variability I mentioned before. So many studies are done in cell lines. And generally, we would assume that if we get a cell line from ATCC, or if we get a cell line from the laboratory next door, and they say it's HEK cell line or it's HeLa cell line, then that we do the same experiment as someone else who has published a paper with HEK cell lines or HeLa cell lines. So this is particularly true for very widely used cell lines, where, like HeLa, where this here Yansheng plotted the number of publications that cite or depend on these cell lines, and it's many thousands over time. So up to now, it's almost 100,000 publications. Research papers have been carried out using HeLa cell lines, and probably similar numbers would relate to other cell lines that are frequently used in the laboratory. So there is different flavors of HeLa cell lines, and interestingly enough, oftentimes these flavors are actually not named in a research paper. So there's basically 2 flavors: the HeLa proper, also referred to as HeLa S3, and HeLa Kyoto, which was once selected for being adherent. So if you're doing imaging experiments, like microscopic imaging experiments, it is an advantage to have cells which actually adhere to a plastic dish or to a surface, because you know where they are and you can image them, and as opposed to when they float around. And so this was as a variant selected, which is strongly adherent. And of course, we would already assume that since the cells have a different phenotype, adherence, as opposed to not being strongly adherent, that they might be somewhat different. So the aims of the study was to do a cross-lab study addressing biological reproducibility. We would collect cell lines from different laboratories which have the same name. I mean, not the laboratories, but the cell lines, HeLa. And then we would measure how different are these cells in their genomic and proteomic composition, and also from the point of view is if they are different from each other, even though they have the same name, do they react differently if you do a biological experiment? Let's say, so you could say, well, I don't really care about how they're composed as long as they do the same thing, then that's also fine. But of course, when we have biochemical background, you would assume if they are different, that they also will react differently. Okay, so this was the experimental design. So Yansheng is now writing this up. It's a fascinating study. So we acquired HeLa cells from different laboratories, and some laboratories were exceedingly careful in that they grew these cells they got from ATCC. They grew them for certain numbers of cycles in their lab and did experiments, and then they froze them down. And then they continued experimenting, and like a few weeks later, they froze them down again. So we have basically a lineage of how they evolve, if they evolve in the laboratory, and some were collected from different laboratories. And then Yansheng measured the copy number variation. It's basically how distorted is the genome. He measured mRNA expression, steady-state protein, proteome turnover, and then a reaction of the proteome to the cell to perturbation, which I don't going to get into. So immediately we can do very broad phenotype. That's how quickly they grow, and they don't grow at the same pace. So this is doubling time and the different strains, and we immediately see that the cells do not grow in the same medium, in the same laboratory, with the same doubling time. So this is already indicating that they're probably not exactly the same. And you will also point out that doubling time might be a phenotype that's selected, is actually selective pressure, because when you were doing experiment, you might want to go with those cells which grow quickly, or those who grow quickly overgrow eventually the culture. So anyway, these cultures grew in different pace, and this is simply the representation of this graph in terms of doubling time. This is data from copy number variation. So each one of these cells has a genome, but the genomes are highly corrupted. These are tumor cells. And so there's translocations, there's deletions. In a normal cell, we would have basically a diploid state that would be from each copy, except the if it's a male cell, which this is not, the Y chromosome would be, of course, in one copy. But so for the other chromosomes, the autosomal chromosomes, you would have 2 copies per locus or chromosome. And this is not true here. There is large distortions of massive duplications. So we talk about this as copy number variation. So specific loci are present in different numbers of copies in these cells. So going from 2, as you would expect, down to 1, then you talk about the deletion, or you would have amplification, higher numbers of chromosomes, entire chromosomes duplicated, or certain sections. So this is true for both the HeLa and the Kyoto cells, and this is basically a graphical representation of a lot of data. These are the cell lines, the individual cell lines. Each circle is one cell line. Each segment is one chromosome, and the heat map indicates the number of copies for a particular locus. And we can already see that simply at the genomic level, there is quite a bit of variance between these cells. We see here, for instance, areas which are obviously reasonably hot here, and in these cells these are cold, and we see very hot here. We see various patterns which clearly says that even though these cells have the same name, so people would think that they do the same experiment. If these cells, they're not actually doing the same experiment because the cells have a different genotype. So now we can group them and relate these cells to other cells, and so we see that the original, the CCL2 cells, HeLa cells, are grouping quite close together, relatively speaking, and they look like cervical cells. So this is where they actually came from in this original patient. But that these HeLa Kyoto cells group quite far away from these and look more like other tissues. So we see that the fact that these cells were selected to be adherent moved them to quite a different appearance based on molecular markers. Then Yansheng analyzed from each one of these cell types, these cell lines, he analyzed the proteome exactly using the techniques that we learned here this week, and he identified about 4,500 proteins, about 5,000 proteins in the cells consistently throughout. And then you can do clustering. You see these clusters appearing, and there's again the Kyoto and the other groups are clustering together, are apart, and then within each cluster is also quite substantial differences. So I'll come back to what this means then, just now enumerating what was done. We have copy number data, we have transcript data. This is proteomic data of fairly high quality. And then Yansheng also measured using a basically pulsed SciLac experiment, the degradation rate of this protein. So basically how long do they live? And we can do this, of course, by stable isotope labeling at a certain pulse. And we can ask how long does it take till the old protein is decaying and the new protein is coming up. And from that, with some formalism, you can identify or calculate the decay rate of the proteins. So this is a summary of this data. Copy number variation, protein, mRNA, and degradation rate. And what we see already, that the patterns look quite dissimilar. So we would, if we had a naive expectation and say we have more of a certain locus or a certain chromosome, let's say this is duplicated, or we have, instead of 2 copies, we have 3 copies, you might naively assume that every transcript is upregulated by a factor that corresponds to the copy number increase, and the protein would be increased in the same way, by the same factor. And this is not true. We already see here, and the degradation rate, that there is clearly not every protein has different degradation, so this is not unexpected. But we also see that some blocks here, which are very distinctively hot or cold from the copy number variation equation, they are analysis. They are at the transcript quite nicely carried over. This is dark. This is cold. This is cold. Here it's much less cold, and here it's also much less cold. So it gets much more diversified. Now this is simply a graphical expression, but now if you plot this on the level of the genome, so this would be chromosome 1 to the last chromosome, and these blocks now indicate the copy number variation. We see at the mRNA level, we see certain blocks like this one here, which is highly amplified, and we see there's a lot of RNA upregulated. And if you go down here, this upregulation at the mRNA level is to a large extent buffered away at the protein level, and so we can follow this for various types of blocks. So the message here is that whatever happens at the genomic level does not just affect the expression of these molecules in a linear and simply transformational factor, but there is regulatory mechanisms available which each one locus has a specific characteristic behavior, how the increase or decrease number of copies of that particular gene are translated over from the transcript into the proteome. And so these cells, even though they have very definitive genome alterations, it'd be genome copy number alterations, it'd be very, very difficult to come up with an equation that says, well, this locus is up 3-fold, therefore the protein has to be up 3-fold. This is clearly not the case. So Yansheng investigated this, and with the hypothesis that it is not just the abundance of the protein that counts, but it is, or another, I should phrase it differently, the hypothesis is that the context in which this protein operates determines to some extent its abundance. So in other words, to the degree by which copy number variation is translated into increased or decreased proteins is buffered to some extent at the level of the protein complexes in which this protein operates. So this is what we like to show here. So we show now a correlation analysis across these 14 cell lines, and we do various comparisons. So here this is copy number variation to mRNA levels, and we see that correlates reasonably strongly. So if there's more copy numbers, then there's more RNA. This is same for proteins. It's not as strong, but it's positive correlation. And this is for degradation rates. It's negative correlation. So this is simply the starting numbers. Now we group this into this total number of RNAs and proteins into 2 groups. Those which are in a complex, known to be in a complex. For instance, we assume or we know that these proteins are part of a complex that is designated as a complex in quorum database, or proteins which are not known to be in a complex. This does not mean that they never are in a complex, it's simply not known. So in reality, the difference that we see would be probably amplified because we include in the complex out proteins, certainly those, some that are also in complex, but we don't know that. So now what do we see? So we see here that when we look at the mRNA level, the copy number variation to mRNA for transcripts which eventually find themselves as proteins in a complex, and those who do not find themselves in a complex, there is virtually no difference. So the correlation between copy number variation is not affected at the mRNA level, and this is plausible because how would the transcript know that it codes for something that is eventually in a complex? But in the protein level, there's a distinctive difference. So this is the overall correlation. We see proteins that are in complex in proteins are much less strongly correlated with copy number variation, and those which are out are more strongly correlated. So this means that the context within which a protein is operating in the cell, namely in the context of a functional module, a protein complex, provides a buffering mechanism that compensates for extra protein being generated. So if you have a copy number variation, let's say 3 or 4 copies of a locus, and this makes more RNA, it probably makes more protein, but this protein has no way to go, so it will not be in the cell. And why is it not in the cell? Because it gets degraded. So these are the degradation rates. So this would be the protein degradation rate for all of them. Protein degradation for the complex in proteins, which is up, substantially up, compared to all proteins. And those which are degradation rate for proteins out, it is lower. So clearly what we see here is that a compensation at the level of how the proteome is organized in the cell is a buffer that determines how changes in the expression, either through copy number variation or at the transcriptional level, is buffered at the protein level. And proteins which are in complex tend to be buffered down strongly through increased degradation. So now, I want to stop with this example here. Yan Sheng then also showed that these cells not only are different in the way they're genomically are constituted, but also and how they are how this genomic variability is translated into transcriptome and proteomes, and covered some rules. But he also showed that when you subject these cell lines to specific biological stimulus, that they also react differently. So the summary of this experiment is that I posed the question, if 2 people do the same experiment with a cell line, do they do the same experiment, or can they expect reproducible research results? And the answer is no, because these cell lines are inherently quite different. In fact, even within relatively few cell cycles from this group that was very careful and froze down cells every few divisions, the cells already evolved in culture and reacted differently. So what this means is that this has huge implications for reporting research results. If 2 people used different HeLa cells and did exactly the same experiment, they might not necessarily do everything perfectly done. They would likely get somewhat different results. And then they might argue in a meeting for hours and say, you did something wrong because your result is not like mine. And the other might say, well, you did something wrong. And in fact, they did everything right, presumably, but they had different starting positions. So I think this has huge implications for reporting research results, because we would not only specify we used HeLa cells or HEK cells, but we would need to somehow also molecularly characterize them. Maybe add a proteome, maybe add a transcriptome, or whatever the case may be. But otherwise, it is not the biologist's fault that the results are not reproducible. They do everything right, but they do basically a different experiment. When then come back to this biological variability. So to finish up here, what I try to show, that reproducibility of research is essential for experimental biology. It makes only reproducibility makes it credible, makes it a science discipline, and it is clearly allows us to discover new biology if we can do measurements across cohorts like this BXD mice or other genetic reference panels, reproducibly we can detect signals in a noisy environment that are biologically meaningful. We think I tried to show that this SWATH, the AA technique you learned this week, is a well-benchmarked technique, and it can generate quite, from a proteomic point of view particularly, highly reproducible research results. And I think they're actually minimally as equally reproducible as transcript data, except, of course, we cannot see the whole proteome, whereas transcriptomes can see the whole transcriptome. And then reproducible measurements are important, but by no means the whole story of reproducible research results, which I try to show with this HeLa example. Even if you do them with a perfectly reproducible and benchmark technique, an experiment that is actually not the same, then, like the other person, then you get different results. So with that, I would like to thank you for your attention. I wish you best success with your research. I hope you can use what you learned here to generate amazing results, and wish everyone a good travel home.
Great, thanks very much, Ruedi. Are there any questions for Ruedi yet? Or is everybody exhausted?
Tired out.
Okay, so Timo.
I mean, I was pretty amazed what you've shown from the Yang Shen's data here. Of course, this will have a bit of an implication for predictive medicine and all these kind of things, how you imagine it in some way. So you have mentioned that there are a number of protein complex components that are kind of counter-regulated in some way. I don't know, do you have any numbers? How many proteins complexes are affected in some way, or how many protein complex components in some way? And also, so you explain it in a way that I understood it, that protein degradation is one way how to balance this out. Yes. But is there also a counter way, so that proteins stay up in expression and then the other protein complex components go also up? So is there different types of co-regulation going on? And is there multiple ways how then the cell lines are going to be in some way messed up?
Yes, so I think this is definitely the case. So this buffering at the level of complexes through degradation of basically superfluous enzymes, protein produced, is simply only one way to that affects the quantitative translation of variation, copy number variation, into expressed molecules. It's definitely there's a clear transcriptional effect, there's maybe translation initiating effect, but it's something that was measurable by the data, because we have the data and we have some base background information, prior information to actually compute that. And it's a significant factor, but by no means the only one, maybe not even the biggest one, that explains the discrepancy between genomic variation, transcripts, and proteins. So I think this is actually a really, as you alluded to, precision medicine, and I think this is an extremely important question, because it's very, very easy now to measure genomic patterns, copy numbers, also transcript measurements on biological or clinical specimens, especially in the field of oncology. And then one tries to make extrapolations on how this affects the physiology of the cell. And I think what this data show, and Yan Sheng has actually done other studies with a rare case where an extra chromosome, extra chromosome 21, was placed in the context of twins, so at samples from otherwise identical twins, where one is trisomic in chromosome 21 and one is not, as normal karyotype. And there we see exactly the same effect, that this extra chromosome has a number of genes, and how the expression of this extra gene is translated into transcripts and proteins is basically regulated differently by each locus. And there's some general rules like buffering at the complex level, but there's other rules, like, for instance, where does the protein go in the cell? So I think there's a lot to be learned about these things, because you can of course say, well, a transcript is up, so it's significant, but if it's buffered down because it has no other complex components to bind to, then it will not have a phenotype. presumably.
And I mean, what you measure is now kind of the abundance relation between complex components, but is this just one link to functional outputs? I mean, there may be many others, of course, that you not
Yes.
derive in the studies, of course, but they're nevertheless true.
Yes. So I mean, I firmly believe that what really carries out functions is complexes. I mean, functional modules usually consisting of more than one protein. It's not so that proteins just float around and do stuff. They're organized functionally, and I think that's what we like to explore. And this is an initial glimpse that this organization actually matters from the point of view of gene expression, but probably also functionally.
Other questions? So I guess we better get to the feedback. Thanks, Ruedi. So this is the last item. If you could take 10 or 15 minutes to fill out the feedback. So I just sent an email to everybody with a link to the feedback form. But if you're not on email, you can get to it through this URL. And if you need to leave right now, then we'd appreciate you can just fill this out later on. Thanks very much.